Hello and welcome to this Jane's Lab video tutorial on handling and processing of fluorescence images. What we're going to do today is take two sets of images from a control cell and an experimental cell, 4 color images, and overlay them in image J, put scale bars on, crop them in Photoshop to get a publication quality image. We'll take these 8 images, open them in image J, tile them so that we can see them all on top of one another. And then the first thing that we were going to do is arrange the fluorescence channels, bright field channels, into a single composite image from these raw individual images. There are 2 groups of samples here. They all look like they're in the nucleus, but they're measuring different things. So we'll go to Image, Color, Merge Channels, and now assign false colors for these grayscale images. The red will be the 555. We'll do the control first. The green will be the 647. The blue will be the DAPI. And the gray will be the DIC, EGFP. You can go up to 7 channels in composites in image J. Click OK. The original images disappear, and now we have a composite with the 4 channels overlaid on top of one another. We do the same thing for the experimental sample. Merge channels. You'll note that I have keep source images off so that those images disappear. You could click it and keep it on, but for now I want to get rid of them. All right, here are now our composite images. You can save these as a composite multi-layer TIFF. It's a separate file. It retains all of the raw original information, and in many ways it's easier to go back and forth handling these as composites. I'm not going to save them because this is just a demonstrative example. Now with these, we can adjust color by image, adjust brightness and contrast. But the thing is that with a multicolor image, the brightness and contrast does not know which channel to apply that scaling to. We will then use the channels tool here to determine which images we want to adjust the scaling and contrast for. So we'll go to color, and now it's only the red channel, or only the green channel, the blue channel, the grayscale, and so forth. Our interest here is in the green channel. This was the experimental measurement that we wanted to have, so I'm going to do the same thing over here, change it to just the color display, change it to the green channel. And then the key thing here is that these 2 settings need to be identical. Right now, they are the same, but perhaps they're not optimal for what we want to do. So we can move the slider to make it brighter or dimmer. The other way to do it is we can set minimum and maximum range. And so often what I'll do here is pick round numbers for the scaling, and then it says propagate to all other 4-channel images, or propagate to all other 3 channels of the image. This is very powerful. If you have many of these that you're doing concurrently, I can click this, and it should do the same adjustment on the other image. Okay, there. And so I hope you notice that the green adjusted here. If I click over here, you can see it also is at 500 and 300. So maybe that's not, there's a little bit of background here. So if I make it 600 and 3000, propagate to all other 4-channel images. Okay, let's say that I'm happy with that. These likewise in the red channel here, you can set to round numbers, 350, 3000, propagate to all other 4-channel images. There, now they're appropriately scaled, and we'll keep it like this for simplicity. if we're happy, assuming that we're saying we're happy with the DAPI exposure, this is a qualitative stain, we're now ready to do a composite. So we can have the composite here. Maybe I don't want to have the DIC image, I just want to show the 3-color red, green, blue image. Do the same thing here, color, composite. You do this after you've done the scaling. These two are now your final merged images, but the problem is that this composite only can be viewed in ImageJ. We need to get an image that can be handled next in Photoshop for cropping or other types of displays and so forth. And so what I'm going to do now is turn this composite into an RGB 8-bit image. Image, color, stack to RGB. Now this is a one RGB TIFF shown here. The same thing here. Image, color, stack to RGB, RGB image. For the purposes of demonstration, I will now take these all away and just have a grayscale individual image. So for this, it's an image type let's for now flatten that to an RGB also, even though it's originally a grayscale image. And then same thing here. Okay, so now we have these 4 images. Let's put a scale bar on. The way that we have this done is that on the lab server, there is a folder called Microscope Calibration. I have it on my computer. You're welcome to grab a copy of it. Here. You'll have to find it on the server, but actually maybe I can just find it on the server. Microscope Calibration. And so we're here, our raw calibration images that you don't need to worry about, but it's these ROIs here that tell you where what the scale bars are for these microscopes. And so I can go back to ImageJ, and now we're going to do analyze let's see. Tools, ROI Manager. Okay, and these are places to do segmentation and so forth, but we use it also to generate scale bars. These images were collected on the 40x oil objective at 2x2 binning on the upright microscope. And therefore what I need to do is open from the microscope calibration the 40x oil 2x2 binned ROI image that indicates 20 microns. I open this and then highlight it on here. This is 20 microns. So I can select this, and if I want to mark it, I can just click delete. And now I have a white scale bar on the black background. Let me save each one of these. Save as TIFF, no compression. Composite, DIC number 1. DIC number 2. RGB, number 1. RGB, number 2. All right. When I'm doing this, I always keep these composites and images open so that I can go back to them and resave as needed. Also, when doing any of these handling of the data, you need to make sure that you are not manipulating the original images. Note that the original TIFFs are already gone. I have these composites, which oftentimes I will save for ease of use. And then these RGBs are also saved as a separate file, separate label, so we can go back to them as needed, and we don't destroy the original data. Now I can open in Photoshop. It's 4. All right, so here are our 4 images. Oh, let's say also that we want to crop these images so that they're not rectangular anymore. We want to make them square. Now there are different tools. There's a cropping tool over here, but I would recommend not using this for this type of cropping that we want to do if we wanted to make it square. And the reason is that it will resize the image based upon whatever is set up here, and that resizing can create or destroy pixels using the cropping tool. And so for the way that we crop to not create or destroy information is by simply changing the canvas size. For example, if I want to crop, let's say we're going to crop it, I'm going to intentionally crop out this scale bar. So we go to here for the DIC image. If I go image, canvas size, there's also a shortcut for this. All right, this tells me how many pixels wide and high that it is. If I want to make it a square 500 by 500 pixel canvas, it's smaller than what it is right now. On the bottom here is where the anchor is going to be. So right now it's smaller both in the width and the height. If I want it to be symmetric all the way around, it's going to prune those pixels all the way around from the middle anchor point. If I put it over here, it's going to pull exclusively from the right-hand side, top and bottom. Here it will crop exclusively from the right-hand side and the bottom. So let me do this to show what happens here. So pay attention to this image here. It will warn that some clipping will occur. There, and it cropped upwards. If I undo that and go back to the canvas size and go 500, 500 in the middle, it will warn again. And now that you see that the image has moved and there's a different set of cells that are being shown there. Let's say that I like that middle image better for this one. So I'll crop 500, 500. Okay. That's the one I want to use. Now to have the RGB overlay be exactly the same thing, all I need to do is adjust the canvas size, 500, 500, so exact same size, exact same anchor point. It was the exact same image. It's just 2 different displays of it. Warns again, and here, hopefully you can appreciate that they are identical and overlapping. Do the same thing here, but instead, because I want to crop out the scale bar, I'm going to crop to the left. There, and now you see I've lost the scale bar. Do the same thing for this. Crop 500, 500 to the left. Okay. Similarly, you can rotate it if we want to rotate it 90 degrees, flipping horizontally or vertically. Sometimes you'll do this so that the scale bar is in an appropriate place that doesn't obscure the key features of what one wants to show. I think I've covered all of the cropping here. What I need to do is now save this. If you want to save this as a crop file, save as to not overwrite. Crop. TIFF, keeping it as TIFF because that's no image compression. Don't worry about these defaults are fine. Save this. I'll note there's a shortcut for this as well, which I usually will use. Crop. And I do the same thing here. Crop. Crop. Okay. Now, if we want to turn this into a figure, we need to open these images in Illustrator. Open. It's these last four that I just saved. And I'll note that each one of these, a couple of things. First, they're enormous, 6 inches, okay? And the reason why they're 6 inches by 6 inches or 7 inches by 7 inches is because they're at a resolution of 72 dots per inch, low resolution screen pixels. We'll fix that when we make the final file. The second thing that I'll highlight is that each one of these images is in a red, green, blue image format. For microscopy images, we want to preserve the RGB format for as long as possible. Therefore, when I make a new panel in Illustrator that ultimately will become my image montage, I need to make sure that the color mode is not CMYK, but RGB. All of the other defaults are fine unless you want to change some of the dimensions, but I can show you how to change the dimensions in a moment. Click OK. So now this is an RGB image. Everything here now is just copying and pasting. Select all, copy, go over to the untitled, and paste. I'll just get rid of that so I can keep track of which ones I'm copying. All, copy, paste. All, copy, paste. And then all, copy, paste. I'm going to just save this as a, in case something crashes. Okay, so it's saved now as an Illustrator file, 4 enormous images on top of one another. Now, this is not publication resolution. These need to be at least 300 dpi, but right now they're at 72 dpi. So what I'll do is we're going to downsize them to something that would be print resolution. You can do this a couple of ways. If you highlight all of these and simply have your selection arrow here, and I hold down the shift button, I'm holding down the shift button. This keeps the proportions of the image constrained. If I let go of the shift button, now I can make pancake cells and distort the image. So keeping the shift button down is critical here when you're doing this resizing. I'm going to move it down. Let's say I think it should be about that size. You see they're much smaller. The key thing now is look at what the resolution of the images are. Now they're at 480 dots per inch or pixels per inch. And so when Illustrator you shrink or expand images, you do not create or destroy data. And this is the reason why we assemble these in Illustrator and not in Photoshop, which can have the danger of creating or destroying pixels. The other way you can do it is by explicitly scaling by a certain percentage. Here, if I right-click and look at Transform, Scale, let's say I want to reduce it to 20% of the size. You know, here this is like holding the shift button down, 20%. And click OK. You get the same effect. All right, I'm going to zoom in, and I'll get these laid out here. Let's see. Let me show the difference here. You'll see it in this RGB. If I take this file and convert it to CMYK, all of these colors will look much flatter. I don't know if you saw that. You see there's a little bit of jostling here. You see how the blue here became much flatter? All right, and that's as a result of this CMYK mapping. These images, it doesn't look too bad, but for some others, it can be really profound, the difference between an RGB and a CMYK image. And these are collected and assembled in RGB, which is why we want to preserve that. Now what we'll do is we'll get these all lined up. Oh, the other thing, what I want to do is I want to get that scale bar back, and I want to put it in Illustrator. So I forgot I need to open up the DIC image that had the scale bar on it. There. Open that up. that's at the wrong DPI, but recall that I scaled all of the other ones by exactly 20%. Scale 20%. All right, now this image is identical to this image, it's just been cropped. And the scale bar is now still reflective of 20 microns. I'm going to use that in a moment. But first, I want to get these aligned. So I'll use the alignment tool here. I'll vertically align. I'll vertically align these to the bottom. And then I'll vertically align these to the right. Vertically align these to the left. And then nudge these a little bit closer. All right. Now, to put the scale bar back in, I want to have it as a vectorized scale bar because there are publishers that say it must be a certain font, stroke weight or color or other annotation. And the best way that I figured out to do this is to zoom in as much as I possibly can on this scale bar, take the line tool, and holding the shift button down, because I can draw a line anywhere. If I hold the shift, it only goes at 45-degree increments here. And then draw a line, not that one, that is as close as I can to this width. So start the line here, hold the shift button, hold the shift button down, and then I drag a stroke out to right to the edge there. Okay, and now I have this line. Well, I have an imaginary line because there's no stroke weight associated with that line. I have to put a weight on it. So this is what a half-point stroke looks like. That's a little thin by my eye. So maybe 1 point. That's still too thin. 2 looks about right to me. Here's my 20 micron scale bar. I don't need this image anymore. And there's my scale bar. All I need to do now is take this over to here, and I can recolor it white. So I change this from black to white, and now I have this as a vectorized format, which, for example, if I want to make it a 10 micron scale bar, I can simply scale it by 50% horizontally, keeping the vertical. It has no length in the vertical dimension. Now I have a 10 micron scale bar. All right, we'll keep it at 20 microns. Oh, what else can I show you? Here, we can label, control. The fonts that we use are Arial, regular. It's the default required font for most publishers. And this is probably about the right size for this image. Experimental. Align that down here. If I want to get these nice and centered, if it's off center, I can use this distribute tool. Horizontally distribute, and then it will place it right in the middle without moving this one. I use this quite a bit. Those two, distribute, right in the middle. If we want to do a shrunken down version of these and put it in as an inset, or vice versa, take this, transform, scale, oh, I don't know, 20% in all directions. This scale bar, oops, we'll hold for here. Drag it there. Likewise, scale this. 20%. You can tuck it down in the corner. You see it's behind the other image. So I need to take this, right-click, arrange, send to back. There it is in the lower corner. Take this, get out of this layer here. Align to the left, align to the bottom. That one happens to be on top. Now if I want to get a scale bar on each one of these, I should take this, copy it, paste it, scale it by 20%. now I have a 20 micron scale bar for that. Just trying to get these aligned. This one lined up. Zooming in and zooming out. These still are centered. You can just drag them by holding the shift button down so that you only go on those 45-degree angles, like so. And then the last thing is I now have a figure, or something that approximates a figure, on a whole piece of paper. To change the bounding box here, the art, they call it the artboard, what we need to do is change use the artboard tool here. So I click on this, and this will enable me to drag these borders. Like so. Save that. And now if I were to save this, just as an example, as a PDF, desktop, save as PDF. All the defaults are fine here. Now if I open it as a PDF, you can see here's the bounding box, here are my images, and everything looks clean and vectorized. I zoom all the way in and it's sharp all the way through. And this is the advantage of the vectorized image format. Okay, I think that this gives the basics for image handling. What do you do in ImageJ? What do you do in Photoshop? And what do you do in Illustrator? So I wish you happy image processing.